Okay, this is my immunology video for test 1. My name is Taylor West. The topics of discussion for this video will be structures of antibodies, T cytotoxic T cells versus T helper cells, primary versus secondary response, malt and gall M cells, clonal expansion. Antibodies are soluble proteins made by B cells in response to exposures to non-self antigens. These antibodies are made of 4 polypeptides that form this Y shape. They have 2 light chains and 2 heavy chains. This region up here is known as the Fab region. This binds to a specific antigen, as you can see here. The FC region lets the rest of the body know something has happened. This region interacts with effector cells and molecules. Right here, between these two, is called the hinge region. It's a flexible tether that allows movement in the Fab region. There are 4 classes of antibodies: IgM, IgG, IgA, and IgE. These are known as monomers, pentamers, and secretary. The IgG, IgE, and IgD are monomers. So you can look at this as GEDs. It's one of the first things you need to get if you didn't finish high school. So remember the first things: monomers, GED. The IgGs are secreted by plasma cells in the blood and are able to cross the placenta into the fetus. The IgEs protect against parasite worms and is responsible for allergic reactions. The IgDs are a part of the B cell receptor, and these activate basophils and mast cells. The monomers and pentamers are most common in bloodstreams. The IgMs are your pentamers. You can remember pentamer is 5, so M has 5 points. 1, 2, 3, 4, 5. These can be attached to the surface of B cells, or they can be secreted into the blood. These are responsible for early stages of immunity. Secretory antibodies are the IgAs. The IgAs are found in mucus, saliva, and tears, and are even in breast milk. These antibodies protect us against pathogens. Okay, so our cytotoxic T cells and T helper cells. They are part of T lymphatic cells, and they are lymphoid cells. These are found in the thymus, which is located between your lungs. They express a unique antigen binding receptor called T cell receptors. These T cell receptors recognize MHC markers. There are 2 types of these cells: the T helper cells and the T cytotoxic T cells. These aid in immune response. Starting with the T cytotoxic cells, these are cell-mediated immunity, and these attack infected cells. They initiate an early warning system, alerting the cell to the presence of an invader. They display CD8, which are membrane-bound protein co-receptors. These also recognize MHC class 1. These are expressed by nearly all nucleated cells of vertebrate species. These are the most common MHC markers. Okay, so we also have T helper cells. They're called helpers because they guide the behavior of other immune cells, such as B cells. These cells, the T helper cells, give directional cues. Instead of the CD8, they display the CD4, and they recognize MHC class 2 markers. These are expressed by professional antigen-presenting cells and a few other cells during inflammation. There are a couple different categories of T helper cells. There are T helper 1s that regulate the immune response to intracellular pathogens. T helper 2s that regulate the response to many intracellular pathogens. And there are also T regulatory cells. These can inhibit an immune response. T helper cells can proliferate and activate cytotoxic T cells and B cells, which is part of the humoral immunity. It's not moving. There we go. Okay, so now we have primary versus secondary response. This is a part of the adaptive immune response. First, starting with primary response, this is the first encounter that your body will have with a foreign antigen. This response takes a longer period of time, and there is a lag time. The activated B cells differentiate into plasma cells, and these cells live for a short period of time. The amount of antibodies produced during the primary response are low. The main antibody produced is IgM because it doesn't overreact, and also small amounts of IgG are also produced. In the secondary response, this is all subsequent encounters with the same antigen or pathogen. This is where they have adapted to the attack. This response time is faster. The lag phase is short because of memory cells that remain for decades. These memory cells help prevent us from catching many diseases a second time. The amount of antibodies produced rise to a higher level and remain longer. Huge amounts of antibody IgG are produced because this antigen is a repeat offender. Also, small amounts of IgM can be produced. MALT and GALT are secondary lymphoid organs. MALT stands for mucosa-associated lymphoid tissue. This is a less organized system of secondary lymphoid tissue. This is found associated with the linings of multiple organs and systems, such as gastrointestinal and respiratory tracts. MALT defends mucosal membranes, and this initiates immune response to specific antigens encountered along all mucosal surfaces. GALT is a component of MALT. This stands for gut-associated lymphoid tissue. This is intestinal epithelium. This functions to protect the body from invasion in the gut. This is done by strong immune cells such as T and lymphoid cells. Associated with this is M cells. M cells are in the digestive tract. They transport antigens across the epithelium. They have a deep pocket filled with clusters of B cells, T cells, and macrophages. These antigens, transported by M cells, ultimately lead to the activation of B cells that differentiate and then secrete IgAs. These IgAs are concentrated in secretions. And last, we have clonal expansion. First, in the bone marrow, the B cells are occurring. The B cells has a receptor that also becomes an antibody. The B cells replicate, and each daughter cell is different from its parents and siblings. Each will have a different and unique receptor. In the thymus, the T cells have a unique receptor that doesn't become an antibody, and they start to replicate, and each daughter cell will have a unique receptor as well. After this, daughter cells then migrate to the lymph node. Affection occurs in tissues presenting a bacteria. Dendritic cells, which are antigen-presenting cells, go into lymphatic vessels and go up to the lymph nodes, where they present the bacteria to the B and T cells that are presiding there. They activate if they have the right receptors, so the B and T cells that match to the bacteria with the right receptor will be activated. B cells can react directly to the bacteria, but T cells need antigen-presenting cells, such as dendritic cells, to present this bacteria to them. Activated B and T cells start replicating like crazy. Each daughter cell will have the same receptor as their parent cell. so that these daughter cells can also recognize this bacteria that is in the body. Most become infector cells who are ready to fight the present infection in the body, but some will also become memory cells. These memory cells will wait for the next time this infection comes around. So when the B cells start to pump out antibodies, and then some of the T cells will stay in the lymph node, and some will travel to the site of the infection. Won't move. Oh, well, that's the end of my slide, so thank you.